Peace students, welcome to our first lab on muscles. This is a quick introduction to muscles. We're going to go through some histology, we're going to look at the microscopic view of all three kinds of muscle tissue, and then we're going to look at the basic model of a muscle cell, and that's it. Easy peasy. So, what I got right here, this is skeletal muscle. And there's actually a bunch of different skeletal muscle cells here. So let's take a look at some of these skeletal muscle cells. This is one skeletal muscle cell right here. Okay, this is one skeletal muscle cell. All right, that's one. And it's extending this way, it's extending that way. It's really long, so it doesn't even fit on the slide. So this is one skeletal muscle cell. Here's another one right here, another one right here, another one right here, another one down here. And we got like a little piece of one down here, piece of one up here. These are skeletal muscle cells. They are also called skeletal muscle fibers. They're called fibers because they're long and skinny. They're kind of string-like. We had nerve fibers that were long and skinny. The same idea. So these are skeletal muscle cells here. Skeletal muscle fibers. The word skeletal tells us that they're probably attaching to and pulling on bone. Now, not all skeletal muscle fibers are attaching to bone at all times. Think about your face and your facial expressions. If you smile, you're pulling on the skin around your lip. If you're moving your eyes, you're pulling on your eyeballs. And so it's usually attached to bone, but not always. All right, a couple things about these guys that we can see. Number one, we can see stripes. Look at all these stripes here. We don't use the word stripes. We use the word striations instead. So these light and dark things are striations. Skeletal muscle cells are striated. They have these stripes because of the pattern that occurs in them in the way that the proteins of muscle contraction are called actin and myosin, and the way those are arranged creates this striped appearance. Now, another thing we can see here is that there are a ton of nuclei. I labeled some nuclei up here with the number 2, but there's more nuclei. Like this muscle cell right here, I can already see it has 4 nuclei, and the muscle cell is long, it's going to have more. We see so many nuclei, and we said there are 6, cells we definitely see here, and there's a heck of a lot more than 6 nuclei. This is because originally, when you are a tiny little embryo, you have these embryonic muscle cells, which are known as myoblasts. So we've seen that blast prefix before, like osteoblast, chondroblast. Myo means muscle, so myoblasts are muscle makers. And these embryonic muscle cells fuse together to give us these adult muscle cells. And this fusion accounts for 2 things that we've noticed already. We said muscle cells are really long and skinny. Well, their length is a consequence of that fusion. Also, the fact that they're multinucleate is a consequence of that fusion. And by the way, when we say long, they really are long. I mean, your longest muscle cell basically runs from kind of the front of your hip to the inside of your knee. So imagine drawing a diagonal line from like your pocket at your hip all the way down to the inside of your knee, and that's the length of your longest muscle cell, which is pretty long. All right, this is one histo slide that is done. Let's go ahead and do another one. Now, I want you to pause this here. And when you pause it, count how many skeletal muscle fibers you see. Point out the striations in the nuclei. Striations are really apparent here. You cannot miss them. Can't miss them at all. Okay, skeletal muscle histology is done for the moment. Let's do some cardiac muscle. Cardiac muscle is found in the wall of the heart. So the heart is this hollow organ, has 4 chambers. They are going to allow blood to enter. They squeeze to expel blood and send it throughout your body. Your heart has cardiac muscle. It's the only place where it's found. It's not found in blood vessels. It's not found anywhere else. It's in the heart. Cardiac muscle is striated. So I can see the stripes here. I can see stripes here. I can see stripes here. I can see them here. They're everywhere. They're not as beautiful and they don't jump out as much as they did in skeletal muscle, but they're still there. I can see nuclei as well. The nuclei tend to be bigger and rounder than we saw in skeletal muscle. Sometimes there's some white space around the nucleus called a Golgi shadow. But we see nuclei here, and most cardiac muscle cells are uninucleate. You'll occasionally find some that are binucleate, but most of them are uninucleate, meaning they've got one nucleus. So we got a similarity and a difference between skeletal and cardiac muscle. Let's pick up on another difference, actually, while we have a moment here. Skeletal muscle cells, for the most part, you have to tell them what to do. Your brain has to send a signal saying, hey, contract. You don't do this with cardiac muscle cells. They are going to contract on their own at their own pace. You don't have any control over that. So we say skeletal muscle cells are mostly voluntary. Cardiac muscle cells are involuntary. All right, what else can we see? There is something else that is totally unique to cardiac muscle cells. And we see a bunch of these things. So I'm going to put some arrows on one here, and the number 2 is on a couple of them. There's one right there, that little reddish line. There's one right there, that little reddish line. One right there, that reddish line. So that's a really nice one that 2 is on. All of these vertical reddish lines in this view, all of these guys are called intercalated discs. Intercalated means kind of like stuck in between. And these things are in between the cardiac muscle cells. And they contain 2 important structures. They contain gap junctions, which we learned about a long time ago. Gap junctions connect cardiac muscle cells electrically so that the entire heart can contract as one more or less big unit. You need the millions of heart cells to work together and not to work individually. So the gap junctions coordinate their activity. And you'll actually learn more about this in AMP2 when you do a whole unit on the heart as part of the cardiovascular system. Now, the other part of the intercalated discs are these things called desmosomes, which we've learned about already as well. These guys are going to strongly anchor cardiac muscle cells together, prevent them from separating as they exert their force. Okay, wonderful. Let's keep on going. I want you to pause it on this one. Look at the cardiac muscle cells that we have here. Find me some striations, some nuclei, and some intercalated discs. You cannot miss the intercalated discs here. They are extremely apparent and very, very nice. Okay, now we got some smooth muscle cells. This is our final type of muscle histology we're going to look at. We find smooth muscle cells in the walls of hollow organs. So pretty much if there's a hollow organ in your body, it's either going to contain a liquid, so think like digestive tract, reproductive tract, urinary tract, or it's going to contain a gas, think respiratory tract there. And in all of those places where we have gases and liquids, we need to move them. So we need muscle in the wall of the tube to move them. And the muscle is called smooth muscle. Now, I've got 2 views of smooth muscle kind of side by side here. I've got it in longitudinal section where the number 2 is. And so you can kind of like get an idea of the muscle cell. I'm not like perfectly on it right there, but that's basically what the muscle cell looks like right there. It's uninucleate. There's a nucleus. It has no visible striations. Now, it still has the contractile proteins, the actin and myosin, but they're not arranged in the same pattern that creates the stripes that we saw in cardiac muscle and skeletal muscle. Okay, what else do we have here? We also see these guys over here in transverse section. So each of these little circular things here is a smooth muscle cell. Sometimes the nucleus is in there. Depends whether we cut right through it or not. This is actually a slide from the small intestine. So the muscle cells here would help move food and food waste onward. Okay, one other thing about these guys, smooth muscle is like cardiac muscle in that it is involuntary. You don't exert any control over it. Okay, awesome. Let's look at another picture here. I got smooth muscle here. Can you see the smooth muscle cells? Can you separate the longitudinally and the transversely sectioned ones? Can you see their nuclei? You should be able to see all that. All right, awesome. Now, we are done with our histology for the time being. We'll do one more slide in a bit. What we're going to look at now is a model of a single skeletal muscle cell. This is a skeletal muscle fiber right here. Right up here, this kind of smooth looking region, this is the plasma membrane of the skeletal muscle fiber. The plasma membrane is called the sarcolemma. Sarco means flesh or meat. So if you've ever heard of a sarcophagus, same root. Lemma, of course, we met in axolemma and neurolemma. We saw that suffix in those words. That means, lemma basically means covering. So the sarcolemma is literally the meat covering, flesh covering. Okay, around it, we see a layer of connective tissue called the endomysium. So it's kind of a loose connective tissue. We see the fibers in there. It is going to bind neighboring muscle fibers together. You got to remember that there was an endoneurium around the axon. We have an endomysium around the muscle cell. Okay, what else can we see? We can see a nucleus right here. Talk about it in a second. We can see some contractile machinery pieces, these little pipe-like cylinder things sticking out. We'll get to them in a moment as well. Okay, got a slightly different view here. I still see my sarcolemma. Very nice looking. See some nuclear bulges there. Remember, it's a multinucleate cell. Got my endomysium, and the endomysium has been cut away to show us the sarcolemma, and also to show us the end of this neuron right here. This right here is the end of an axon. So I got the end of an axon right there. I got where the 11 is, right there and right there. I got some telodendria. Each of them is a telodendrion. Number 4 right here is an axon terminal. So what I'm looking at here is where an axon terminal, so this right here is where an axon terminal is synapsing with a skeletal muscle cell. The axon terminal is going to be filled with a neurotransmitter, which is known as acetylcholine. When an action potential comes down the axon, down the telodendrion, and gets to the axon terminal, the acetylcholine is going to be released. Now, over here, we've cut the axon terminal away. By the way, axon terminal is sometimes known as the synaptic knob. Your lab manual will call it synaptic knob. Anyway, so when the action potential comes down to the axon terminal, it's going to release acetylcholine. And the acetylcholine is released onto this region of the sarcolemma. This region is called the motor end plate. Motor because, well, we're going to have muscle. End because we're at the end of the axon terminal, of course, right? The word plate is a little misleading. The word plate has the implication that it's going to be flat, but this guy is not flat. It is very ruffled. It goes up and down and up and down and up and down and up and down. And the reason for the ruffling is it gives us more of something. You know this. It gives us more surface area. And if I have more surface area on this motor end plate, I can have more receptors for acetylcholine. The receptors for acetylcholine, by the way, are referred to as nicotinic receptors. I bet you can guess what else they bind to besides acetylcholine. Now, motor end plate is what receives the acetylcholine. More surface area because of the ruffledness gives me more nicotinic receptors. Now, what these guys are ion channels. So when the acetylcholine binds to them, they open up, sodium rushes in, I get a graded potential, then my muscle cell experiences an action potential, and then it is going to contract. And by having the ruffles, I get more receptors, I get more graded potentials. You know that graded potentials can add together and increase the likelihood of threshold and of getting an action potential. All right. This is all good. I think we hit everything I wanted to hit here. That was kind of a lot, I know. Let's take another look. We kind of like zipped it around so we can see the inside. Before we get to the inside, we got our endomysium out here still. We got our sarcolemma as well. We can still see that. A little bit of tape on it. We got a nucleus right there. Over here we got our axon terminal right there, or synaptic knob. You can see that. We can see the motor end plate right there. Another one right there. We see all of its folds, which are there to give us more surface area. Now, by the way, this particular synapse is referred to as a neuromuscular junction. A neuromuscular junction, which makes sense. It's where a nerve and a muscle are kind of joining, not physically joining, but functionally joining. The neuromuscular junction is going to consist of that axon terminal, that motor end plate, and also the cleft, the synaptic cleft between them. Okay, when we look inside this muscle cell, we see all these cylindrical bundles of proteins. Each of these individual guys right here is called a myofibril. They are bundles of proteins, primarily the contractile proteins, actin and myosin. And you can see the arrangement that they have here, the stripes. Those, of course, are the striations. Now, in the lecture part of the course, we are going to get into detail about what exactly happens within these striations, what exactly actin and myosin do in order to cause the muscle cell to shorten powerfully. In other words, to contract. Okay, beautiful. We are done with this. Let's see what else we got. Got a top view here. We can see the sarcolemma on the outside. Right there. Dun, dun. I got a nucleus right there, kind of hiding under that. Got all of these myofibrils. The clumping here of them is nothing. That's just an artifact of preparation when you look at this stuff under a slide. So don't worry about like the grouping here. It doesn't matter. Okay, sweet. Let's go on. I think this is our last slide here. Yep, this is our last one. What we're now doing is going back to the histology. So I have 1, 2, 3, 4, 5, 6 skeletal muscle fibers. I see their striations. I kind of see a nucleus here. I don't really see very many other nuclei, just the way this slide is. But what I do see here is this axon. This guy right here, this dark bit right here, there's an axon. Branching to give me telodendria. So I got a telodendrion that I'm on right here. I can draw an arrow on that guy. There's a telodendrion right there. And I see a bunch of axon terminals. Pretty neat. So I see neuromuscular junctions here, which is pretty awesome. All right, that does it for this video. This is our introduction to skeletal muscle, really to all muscle. Next time we meet, we're going to start going through the muscles of the face, the eyeballs, the neck, and the torso. Then we'll meet again to do muscles that move the upper limb, then muscles that move the lower limb, and then we're done. All right. See you later.